oh, got it. To talk about hope, humans on planet Earth, or long-term impacts on biosphere dynamics. And it's a European Research Council advanced grant that started in 2018 and will end midway through 2023, because we've been given an extension because of lockdown and COVID and all that. And it will probably continue for another year, thanks to the generosity of the University of Bergen. So, John, in his introduction, commented on the importance of setting out the terms that we use. And I come to this sort of topic as a paleoecologist and as an ecologist. And therefore, the terminology that I use is very much from the biological and ecological world. So patterns. These are what we can observe today if we do, for example, an ecological survey. What we can, please move this window away. The processes, these are the underlying mechanisms that generate these patterns. So, as I say, we have modern day patterns, we reconstruct past patterns using historical or paleoecological data. And of course, we can predict patterns in the future using appropriate models. But what we can never do is to observe, unless we actually do experiments, the processes, the underlying mechanisms that have generated these patterns, like dispersal, establishment, assembly rules, competition, and so on. And these are unobservable, and therefore they can only be inferred. Now, biosphere dynamics, it sounds very grand. This is the sort of terminology I had to use in the ERC grant application. These are the ecological and evolutionary changes that occur as biological systems respond to and contribute to environmental change. So at a variety of scales, from population growth rates, shifts in geographic range, compositional change, and then over long time periods, of course, speciation or extinction. And what we can approach this is through ecosystem or ecological properties, the composition, the abundance, the richness, the diversity, the evenness, the turnover, species co-occurrences, etc. So there are, I wish this thing would go away. It says, please move this window away. Does anybody know what to do? Just go away. So, as you all know, then the basis of the Page's Human Traces group is what was the impact of prehistoric people on Earth's biosphere and climate systems? Did, for example, Neolithic people initiate an early Anthropocene through forest clearance, agriculture, burning, and so on, or even earlier, as Bill Ruddiman has suggested, in Asia? And then there is now an enormous wealth of data from paleoecological studies collected over 80 years at least, showing human impact on vegetation composition, extent, soil erosion, water and nutrient budgets, landscape resilience in many parts of the world. Therefore, we know from the very beginning that there has been and continues to be human impact on biosphere patterns from the poles to the tropics. But did these human activities modify the biosphere in such an extent that the fundamental underlying ecological processes shaping communities and biomes were changed as a result? So what hope is trying to do is to test two major hypotheses. That firstly, ecological processes change through time, the last 6,000 years, say, due to human activity, and that the interrelationships or the correlations between these processes have also changed through time. In other words, the ecological processes on Earth today are different from, say, the processes on Earth 6,000 years ago. And these are fundamental questions in Earth system science. So how is HOPE trying to test these hypotheses? Well, it's got three major inputs. There's the HOPE objectives: do ecological processes change through time? We're relying very much on basic pollen and archaeological data, and these come from pollen databases, which rely on pollen analytical studies on human impact and also archaeological surveys. So this is the data input, so to speak. Then we have a series of numerical and statistical tools, analytical methods. Then from our understanding of contemporary ecological processes in biogeography, we then can try to answer the question, did ecological processes change through time? So fossil data, basic pollen data with associated radiocarbon dates from lake sediments and peats from sites across Europe, North America, Latin America. And when I used this diagram from Eric Grimm's article in the Encyclopedia of Quaternary Science, there were hardly any data points for Asia or Oceania. And so the original proposal, we conveniently dropped Asia and Oceania. But now, we now have data from international open access databases, such as the Latin American pollen database, Neotoma, Pangea, and through my very good contacts with Chinese colleagues, we now have the Chinese pollen database, and so we now have harmonized data from 1,781 pollen sequences, and more to come from Oceania and Africa. Now, the analytical tools, we have numerical and statistical methods to detect major patterns in each pollen sequence. And we can estimate, using appropriate numerical techniques, quantitative characteristics of each individual pollen sequence: taxonomic richness, diversity, evenness, beta diversity, rates of compositional change, turnover, and functional and phylogenetic diversity. And these are what I call pollen assemblage properties. And these are a proxy for the ecosystem properties that we're really looking at. And so we have a range of fairly robust and reliable statistical techniques, such as reduced rank multivariate regression, or more commonly known as redundancy analysis, generalized linear and additive mixed regression, and computer-intensive permutation tests to test the basic hypotheses. Now, the ecological processes, of course, we know from present-day ecology that there are extrinsic or exogenous environmental factors such as climate change and human impact. There are also intrinsic or endogenous interactions such as competition, facilitation, and so on, and neutral processes such as historical legacies or random events. And then, of course, the real world, as distinct from the statistical world, these probably all operate simultaneously to varying degrees to generate the ecosystem properties that we can try to quantify from pollen data. So the challenge is to infer underlying unobservable processes from the observable patterns and these estimated assemblage properties. Now, here's a very simple example. It's one of these notes that Gregory Dietl published in Nature in 2015, which actually started my interest in this topic. Here's the concept of species co-occurrences. Do pairs of species occur randomly or non-randomly? So this would be for species 1 and species 2, a random pair, or they might be segregated, they don't like each other, or they might be aggregated and come together. And if one can detect segregated pairs, this would suggest dispersal limitation due to, for example, habitat fragmentation and a high mosaic landscape. Aggregated pairs would be long established assemblages and low mosaic hypothesis. And so we need in this the ecological understanding of the ecosystem properties that is needed to interpret the pollen assemblage properties. So hypothesis 1, ecological processes have changed with time. So here we basically do a form of change point analysis of the observed changes in the pollen assemblage properties and compare that with a random model to test whether the changes are aggregated, segregated, or random, and linked to human disturbance events, climate change, or other factors. And hypothesis 2, the correlations between these processes, we're using multivariate probostase analysis, which allows you to compare different times, 6,000 years, 5,000, and associated permutation distances. And of course, by working globally, at least within the Northern Hemisphere, then we can do tests within and between continents, such as Europe versus North America, Europe versus Asia, and within and between biomes, like the boreal forest, compared, say, with Mediterranean biomes. And with the different spatial scales and the experimental designs which are imposed on us by nature, we can do about more than 60 independent tests of hypothesis 1 and over 100 of hypothesis 2. So we're very much using we're taking advantage of the power of John Platt's concept of strong inference approach, as well, of course, the fundamental aspect of science, Thomas Chamberlain's method of multiple working hypotheses. So why is hope important to Earth system science? Or put another way, how did you sell this to the European Research Council panel on Earth system science? Well, Earth system science, geological science, paleoecology, in fact, all historical sciences rely on Hutton's principle of uniform returns. The present is the key to the past. Physical and biological laws apply to all geological time as well as the present. And that is the underlying logic and conceptual methodology by which the past can be reconstructed and the future predicted. And so this is the basic research approach and philosophy of Earth science, Earth system science, call it what you might. But in 2015, a paper appeared in Nature by Kathleen Lyons et al. And Gregory Dietl wrote a commentary on it, suggesting that biological assemblages after 6,000 years ago were formed in a fundamentally different way from earlier assemblages unaffected by human activity. And they proposed that uniformitarianism should be discarded. And the text that I have in italics that quotes them, that using natural experiments to predict future changes is flawed, and that out is the use of uniformitarianism as a guiding principle in earth science. So therefore, at the stage our science is at, there's an urgent need to assess critically and robustly the view that uniformitarianism should be discarded. Without it, much of Earth system science, restoration ecology, conservation strategies, the future, that are all based on, to some degree, past ecosystems, grind to a stop. And so therefore, hope, I argue, to the ERC was important to Earth system science and ecological science. So what tools do we have? Well, after getting the grant, I got this parcel from the European Research Council, So it consists of a ERC hat, a cap, a notepad, a canvas bag, a badge, and some sticky labels. So that really got the project off to a great start. And we thought, well, we better need a bit more than that. So we also have a team of absolutely first-rate, early, mid, and I have to confess, very late career scientists. There's myself as PI, 4 people as researchers: Suzette Frantua, Vivian Felder, Kuba Bata, and Andre Mortel. University gives us the service of one of their administrators to keep the economy straight, which is very difficult because all the grants are in euros and the exchange rate between euros and Norwegian kroner is changing all the time. 3 co-PIs, Jon Ivy Gytnes, Alistair Seddon, and my wife Hilary Birks, and then excellent technical help from Kathy Jenks, Lynn Kruger, and Al Beeston. But we're not just in Bergen. We then have a fantastic group of external collaborators. Sadly, Eric Grimm, who many of you knew, is no longer with us, but Eric was an enormous help at the beginning, as was Jack Williams, and then working on phylogenetic and functional diversity, Trin Riatulu from Tartu, Sandra Neuge from Southampton, soon moving to Barcelona, and Manuel Steinbauer are closely involved, Henry Hoog-Himstra for Latin America, Thomas Giesecker, and then statistically we get valuable advice from Kayo Brack in Wageningen in the Netherlands, and his collaborator Petra Schmilova. And then various people have or are helping with data: Willy Tinner from Mediterranean areas, Fachu Chen from Beijing and Lanzhou, who really opened up the Chinese pollen database for us, Simon Haverly, who in the Indo-Pacific database, and then Janet Wilmshurst, who will be working with us further on islands. So you could say, and I think at times I would agree with you, that it's a very ambitious, it's in fact perhaps too ambitious a project. We now have this enormous data set, 1781 sequences now harmonized taxonomically with Bicron age depth models and events of human impact detected from the pollen data. A workflow developed in R to handle this gigantic data set, and potential predictor variables are currently being compiled. Paleoclimate model results independent of the pollen data on Chelsea, and with the People 3K project, they've generated for us radiocarbon probability density functions for various different parts of the globe as a proxy for human density. And so the software for hypothesis 1 testing is currently underdeveloped. It's nearly all actually developed. Of course, none of this has been helped by COVID-19 lockdown, working from home, and sick leave for 4 members of the Hope team. But that's not unique to Hope. That's a problem for many, many research groups. So we've published some papers already. Sandra Noga and Manuel Steinbauer led a paper on the human dimension of biodiversity changes on islands in science, and Andre Moffel, Suzette, Alistair Seddon, and Jack Williams led a paper on global acceleration in rates of vegetation change. And then the methodology of rate of change, which we've now refined considerably from what we were doing in the early 1990s, has now been published in Review of Paleobotany Palynology. And I wrote a large review paper, Contributions of Quaternary Botany to Modern Ecology and Biogeography. And then closely related to Hope, papers on modern pollen and planet richness, trying out some of the ideas on compositional turnover and variation in last interglacial Eemian pollen sequences, reflections on the use of ecological attributes, and then a recent paper with my Chinese colleagues on cycles of grazing and agricultural activity during the historical period, and how these related to climatic and social changes in northern China. And in addition to those papers, which are strictly Hope, then of course the members of the Hope team have been publishing stuff not quite so Hope-ish, about 25 papers, mainly in the fields of biogeography or plant ecology. So I just end by acknowledging the Hope team for all their work. Eystein Janssen, who at that stage was head of the Birks Climate Center, and it was Eystein who suggested, when I discussed these ideas with him, why don't you try for an ERC grant? And he specifically said the Earth System Science grant. University administrators helped with all that. And then I had the very good fortune to go with Hilary and Paul Cardy and friends to the Pyrenees in 2016 in the summer. And that really helped to clear the mind. And then when I got back, I was able to sit and write the proposal. Our previous head of department and current head of department have been extremely supportive. And I'm very, very grateful to my good friends, Cathy Willis in Oxford and Rick Batterby in London, who read the first draft of the proposal and helped improve it. So that's what Hope is about. We are hopeful, that it will succeed. Oh yes, and of course the acronym was thought of by Hilary, who's very clever. She has a crossword sort of mind, humans on planet Earth. So thank you very much for listening.